This is the iClicker thingy. We're gonna hide it for now. Okay. As promised, I will tell you today about DNA structure and DNA replication, but I wanted to give you a preview of what we're going to do on Monday, as in why should you bother learning the structure of DNA and how it replicates, both in vitro and inside you and I. Well, the Wall Street Journal yesterday told us that New York law enforcers push for expanded DNA database, you see. Governor Andrew Cuomo's proposal to make New York the first state to require every convicted criminal to provide a DNA sample was endorsed Wednesday by law enforcers. And why? Well, they listed among their reasons the need to satisfy potential jurors who've come to expect the scientific evidence from watching crime dramas. DNA, said Erie County District Attorney Frank Sedita, DNA is undoubtedly the single most important advancement of a generation. Wow. I want to emphasize as strongly as I can that I respect the fact that prospective jurors watch a lot of crime dramas. However, I don't want the fact that this appears on this or that TV show to disparage the quality of the data. The use of genotyping to identify a person is remarkably powerful, sophisticated, and robust tool. It was developed by the federal government, specifically by the National Institute of Standard and Technology, or the NIST, and from John Butler, who was part of that effort, I got this slide. These are the short tandem repeat, STR, loci, which is genetic for positions in the human genome, that can be used to conclusively determine if a piece of DNA comes from a specific person or it does not. Now, before I can explain to you what this is, and there are actually at least 3 marvelous topics that can be taught using the CODIS loci. The first is Mendel's laws. The second is the nature of interhuman genetic variation. And last, of course, the polymerase chain reaction that you are doing this week. But before I can explain to you all of this, I need to teach you what DNA looks like and how it replicates. And once we're done with that, I promise you we'll go back to this and you will be the prospective juror par excellence. You will know all about short tandem repeats and not from dramas from Bio 1A. Oswald Avery and his 2 colleagues at Columbia in 1944 did a beautiful experiment that proved in the short term that DNA is the agent that causes the transformation of benign bacteria Streptococcus to a malignant one. Perhaps malignant is the wrong adjective. A lethal one, how's that? And one would argue that one could have stopped at that. But as I mentioned, Avery wrote a very understated discussion section of his paper. He didn't say, DNA is the genetic material, yay. No, he was like, well, DNA is the transforming principle. We need to study this question further. The issue of DNA being the genetic material, in fact, wasn't settled in the mind of the general molecular biology public until 1952, when these 2 individuals, who are forever in the history of molecular biology, Alfred Hershey and Martha Chase, did the famous blender experiment. And I will walk you through it. The experiment proved that viral protein and nucleic acid have independent function in the growth of bacteriophage. In addition to the science that I will walk you through, and I'm expecting you to know and understand for the midterm, there is a wonderful teachable moment from the structure of scientific discovery perspective. Without, for an attosecond, wishing to disparage the significance of their contribution, I really want to emphasize that among practicing molecular biologists, the consensus is 100.00%. This is a sloppy experiment, and that's a strong word, sloppy. It is sloppy, technically speaking. It's not even close to the quality of data in the Avery experiment. Why is it in every Bio 1A textbook on planet Earth? It was done at the right place and at the right time. That's the issue. It occurred at the right place and the right time in the history of biology. And one of the things I'm going to ask you on the midterm is to highlight, flaw is a strong word, the incompleteness of the argument in the Hershey-Chase experiment. The goal is not to just teach you how to appreciate correct data, but how to visualize flaws. And with that, mesdames et messieurs, bacteriophage, which is Greek for the entity that eats bacteria. You're very familiar with this, so I'll go through this very quickly. Bacteriophages are essentially parasites of bacteria. They attach themselves to bacteria, as you can see here, inject the program for the making of more of themselves into the bacterial body. Then that program reroutes bacterial metabolism to produce more phage. And then those phage are released and infect other bacteria. And then the cycle repeats itself until there are no more bacteria left. Incidentally, an important commercial application of phage today is to remove contamination from meat. So you can take meat and treat it with bacteriophage, and the pathogenic bacteria that may or may not be on that meat will be killed using this process. There are two lines on the slide, which has all its text removed from the textbook, because Hershey and Chase decided to ask whether the genetic program being injected is composed of protein or DNA. Now, how did they do that experiment? They used a radioactive label. In fact, today you will see two experiments, one by Hershey Chase and the other by Matt Mieselson and Frank Stahl, that both used radioactivity. And one thing I'm going to teach you and then ask you on the exam is, why was radioactivity useful? And in fact, as you will see, it was useful for different reasons. And I'm expecting you to know both. In fact, both of those reasons are in use in biology, and not just biology today. In brief, Hershey and Chase both knew that there is a specific element, and in a remarkable bit of pedagogical concurrence, the periodic table of the elements is right there, illuminated. There is a specific element there which is only found in protein and is not found in nucleic acid. That element is sulfur. Et voila. Conversely, the element that is present in nucleic acid is in fact present in very trace quantities in some proteins, but in completely trace quantities. So what is that element that distinguishes nucleic acid from protein? Phosphorus. Very good. Both of these elements exist as radioactive isotopes. The purpose for Hershey and Chase of using them was you can track them. You can measure how much radioactive sulfur your solution contains. It's a 20-second test, and it's quantitative, scalable, and robust. In other words, if you grow bacteria in the presence of S35, which is a radioactive isotope of sulfur, you can measure how much entity, whatever the entity happens to be, your solution contains, because you can quantitate the amount of radioactivity. Similarly, if you grow bacteria in the presence of a radioactive isotope of phosphorus, P32, by the way, this experiment was done in 1951. Every molecular biology lab on Earth, to this day, uses these two elements daily. S35 and P32 are in routine everyday use today. So you're not using archaic technique, you children of the age of the iPhone. You're learning modern day technique. You grow phage in the presence of P32, and you get phage labeled with P32. Hershey and Chase realized then that they have two distinct preparations. One in which the protein of the phage is labeled with S35, but not the DNA, and one in which the DNA of the phage is labeled with P32, but not the protein. And then they could ask, which of the two radioactivities entered the bacterium? Whichever one enters must be the program. So let's walk through the interpretation first that every textbook on planet Earth contains, and then briefly look at the primary data. The interpretation of the data that your textbook teaches you is that if you take a phage that looks like this, in which the protein is labeled with S35, there's no radioactivity within the cell, and the S35, therefore, is in the phage coat. Conversely, when you do P32 labeled DNA in the phage, you infect the poor bacteria, you get P32 within the cell and not in the phage coat. Ta-da! The entity labeled with P32 has entered the cell. It must be the genetic material. DNA is genetic. Rock and roll. Not so fast. Here are the primary data. Take a look and see for yourself. Do you accept this black and white conclusion? So first, let me point out that they're not actually measuring amount of things inside the bacterium. They're measuring the amount of things that have stayed outside the bacterium. The x-axis is time. Phage infect things very fast. How do they determine what's in and what's out? The phage, after all, do attach themselves to the bacteria. This is where the blender comes in. They basically attach the phage to the bacteria, and then after a defined number of minutes, press the blender button. And the bacteria go, wee! And the phage just falls right off. And then they precipitate the bacteria and ask, what's left in the supernatant? I realize I'm going a bit too fast, and some of you have this, they what? I'm going to walk you through this. I'm going to walk you through this once, and please then read the textbook over the weekend. And I promise you by Monday you will understand. All of the bacteria are infected near immediately. And then they ask, well, does all of the DNA enter the cells? Well, 30% of the DNA doesn't. Why? Well, it just didn't. We don't know why. Furthermore, did none of the sulfur enter the cells? Well, 20% clearly did. In other words, a good chunk of the nucleic acid, the supposed genetic program, didn't enter the cells, but perhaps even more concerningly with respect to the conclusion of the experiment, 20% of the protein appears to have gone inside the bacteria. Compare and contrast with these statements. What's going on here? The fact that we know that the conclusion from the experiment is true has colored our perception of the data. We want the data to say that no protein entered the cell and that all the DNA did. That's not what the experiment said. Should some of you, and I'm hopeful that's the case, choose to become experimental scientists, think back to this example of how what you wish to show can bias your data interpretation. This doesn't mean that you shouldn't draw conclusions, but this does mean that you should take your data with a grain of 5 molar sodium chloride. Anyway, DNA is the genetic material. Enough already. Fair enough. Now that this little module is complete, let us click. Please pull out your clickers. Please turn them on. We will first have a test drive question, but before I do that, all right, give me a second, make sure that your clickers are on. So in principle, when I click this button, a little screen should appear, and your answers will materialize and will be recorded, and I know who said what. Let's test drive the system. All right. Take a look at this image. The question to you, this is not for points, this is to warm up the clickers. What do you think this is? The options. This is the winning image in the 2011 Draw Like Salvador Dali contest. B, this is a cartoon depicting the bubble that US lawmakers work in. C, this sphere shows how much water there is on Earth. D, this is the size of the Chicxulub asteroid that, as all of you know, of course, hit the Yucatan 65 million years ago and caused the extinction of the dinosaurs and the onset of mammals. And last but not least, this is actually quite simple. This is the size of the moon relative to the Earth. Okay, do you understand the questions? Think for 30 seconds about what you think is going on. All right, now in principle, here goes, folks. When I click this, start polling. Whoa. No, well, I have all these answers being registered, so. I am told that you should point it at the, like, engage. All right. 541. Wow, thank you. Thank you for being here in such impressive numbers. Okay, 545. Last one, make up your mind. The answer is 42. Come on. All right. 60 seconds have elapsed. Let's stop. Let's look at the data. What did you say? All right. So 4% said A, 18% said B, 11% said C, 22% said D, and most of you, in fact, half of you think that this is the size of the moon relative to the Earth. Well, this is good. Brace yourselves, you guys. This is the work of Dr. Adam Neiman, and it's conceptual computer artwork of the total volume of the Earth's water, seen as a sphere centered over North America. The Earth is that fragile. There's this little water on Earth. If you gather all of it, it'll form a sphere that's only 1,390 kilometers across. We have limited resources. It gets worse. Let me show you what the atmosphere looks like if the entirety of the Earth's atmosphere were generated into a sphere. I know. The reason that you're saying, why did you do this to us? To remind you that the Earth is fragile and needs to be protected. And it's your generation's responsibility to do that. All right, and now the real clicker question for 1 point. Theodore Bovary used these shebangis to. Whoa, where did you go? Come back. No. Oh, good Lord. Aha. Prove that the cytoplasm has no role in heredity. Prove that a bastard sea urchin can be generated. His words, not mine. C, establish the role of the nucleus in heredity. D, establish the feasibility of growing haploid sea urchins. And E, work out techniques for sea urchin egg enucleation. Okay? Go right ahead, please. Honestly, I'm happy to cancel this point if it doesn't work or you think you weren't legitimately tested. And what we'll do is, okay. All right, this is good. Let's take a look what's going on. All right. Good job. Nicely done. Good for you. Now, notice that I deliberately wrote, I mean, other than A, his experiment didn't really address the role of cytoplasm. I mean, all of these came out of Bovary's experiment. But I'm glad that you all so unanimously correctly interpreted what's going on. His goal was to show that the nucleus drives cell phenotype. Okay. Now, since you have the clickers out so we can put them away, is it okay if we do the office hour thingy? That way we can just decide. So there turned out to be a challenge with respect to getting a room that I could use. So I got 180 Town Hall, which is right there. Your options are Monday 9 to 10, Wednesday 9 to 10, Friday 9 to 10. So right after class. Alternatively, I really am planning to come to office hours, but none of the time slots listed work with my class schedule. And of course, there's always office hours, schmoffice hours. And I promise you, I swear to you on the memories of Gregor Mendel and John Lennon, I will not look up the names of the people who said office hours, schmoffice hours. I promise you, on the most sacred names I can come up with, to me as a Beatlemaniac and geneticist. All right. Folks, the students who I see sitting next to me having trouble with their clicker, part of it has to do with batteries. So, okay, let's see what you think. Well, so this is actually, so the 100 or so of you that cannot come to that time window, I promise every week I will generate a slot where it probably be Tuesday or Thursday late in the afternoon where you can come talk to me if you want to. And the 50 of you who don't want to come to office hours, I'm just kidding. Thank you for your honesty. Okay, what I'm probably going to do is do one on Wednesday and one on Friday. And then for the folks who cannot make it, I'll work out some other time. So there'll be one on Wednesday and one on Friday. All right. Feel free to put your eye clickers away. All right, enough of this silliness. Let's firmly enter 2012. Before you is a string of letters, parsed into words, and put together into an English sentence. Where is it? It's right there. Why am I showing you this? This is the sequence of a human gene, RPE65, mutations in which cause congenital blindness. And I showed you last time data from Jean Maguire's clinical trial on the use of gene therapy to cure Leber's congenital amauropathy. It is also a string of letters. It is also parsed into words. And these are also put together into a sentence, a biological one. It's marvelous, isn't it, that to a geneticist, the string of letters above is as legible as the slide title is to you. And what I'll do over the next 4 lectures or so is teach you how to read genetic. Now, I completely appreciate the fact that at this point, this has sort of, there's no fundamental difference as far as you are concerned between this and this. But as you will recall in that silly movie, there was a character who could actually look at this and actually understand what's going on in the matrix. So by the end of the section of 1A, you will be able to look at the genetic code at the bottom and go, ooh, alpha helix. A practicing geneticist looking at this picture would immediately understand that this says start A, G, C, T, end. How is that done? Well, the letters themselves have chemical identity. Just like the cognate letters, G and C have semantic identity. Your visual cortex, which is right there, has no difficulty looking at the letter G or looking at the letter C and realizing there is a little squiggly, and therefore it's a different letter than this one. Similarly, Mother Nature has no difficulty looking at this heterocycle, which happens to be a purine known as guanine, and then looking at this heterocycle, which happens to be a pyrimidine known as cytosine, and going, oh, A, they're different, B, I know which one is which one. There's no fundamental conceptual difference between the ease with which you tell a G from a C and the ease with which Mother Nature determines that this is a G and this is a C. And how that works, I'm going to explain to you in a second. Similarly, just like the letters A and T are immediately parsable by you, well, that's the A, that's the T. Mother Nature looks at that heterocycle at the top, which of course is a purine known as adenine, or at the heterocycle at the bottom, which is a pyrimidine known as thymine, and goes, you're an A, you're a T. Next. There's 4 letters. So much can be said with them. I'm about to explain to you one of the coolest and deepest concepts in genetics, which is that DNA has directionality. The chemical basis of that directionality, in fact, does not reside in the letters. So A, C, G, and T do not have a left or a right from Mother Nature's perspective. It is the way they are strung together. And they are strung together with each nitrogenous base being attached to a 5-carbon sugar, a deoxyribose. Now, of course, as all of you learned in high school, the term deoxy comes from the fact that that carbon atom has a hydrogen attached to it and not a hydroxyl. RNA, of course, is the molecule in which that's a hydroxyl, and as a consequence, RNA is a lot less chemically stable. And the importance of that, for the fact that you and I are alive, cannot be overstated. The 5-carbon sugar is not a perfectly symmetrical ring. It has 5 chemically distinct carbons, which for historical reasons are known as 1', 2', 3', 4', and yes, you guessed it, 5'. Individual nucleotides are strung together with a phosphodiester bond, a term you all remember from chemistry or whatever cognate class you took that would have taught you this in high school. The phosphodiester bond is formed between 2 chemically distinct carbons in the ribose. Specifically, the 3' and the 5' carbon. Let me restate this slowly, because if you understand this, then everything else will be sitting under the campanile and sipping some iced water. If you don't get this, it will be harder. In the string of text that is our genome, letters are held together by a chemical bond. Here's ethane, not the chemical bond in our genome. Ethane, yawn. Note that the 2 carbon atoms are chemically indistinguishable. There isn't a left or a right carbon. This carbon-carbon bond in this molecule has no directionality. In striking contrast, the bond between 2 nucleotides links carbon 3 of one sugar to carbon 5 of the next sugar. Therefore, the bond is directional. In other words, if we were to think about the way nucleotides are daisy-chained to form a single strand of DNA, they're not daisy-chained like this. It's sort of the walk like an Egyptian. This is an obscure pop culture reference from the 1980s. None of you were born there, fortunately. The nucleotides are strung together like this, as opposed to like this. This means there's literally a front and a back, which in chemical terms is known as 5' and 3'. And there it is. The 5' end and the 3' end. For reasons I'll explain on Tuesday, DNA, for purposes of communicating information, is read in the 5' to 3' direction, which is why to a geneticist the string of text is start 5', A, G, C, T, end. And I chose that for a reason, because there we go. Start, A, G, C, T, end. So when you stare at this 300 nucleotides of genetic text, please understand that every letter is a nucleotide, and the nucleotides are strung together directionally, such that there's a start and an end. And by looking at any individual nucleotide, for example, this one, we can immediately determine where the 5' and where the 3' end is. Et voila, the completely annotated figure. What would I like you to know about this? I'm sorry to say everything. You just have to. It's just part of education. But it's wonderful. I promise you that all of these polysyllabic words will shine. Download the slide of BSpace. Make 2 copies, one with absolutely no text and one with lots of text. And then practice taking the one with no words and populating it and then comparing it to the backup copy. And do that until you succeed. You need to know what purines and pyrimidines are, which are the purines, which are the pyrimidines. Do you need to be able to draw the structures from memory? Absolutely not. But you do need to be able to recognize everything here. Where's the 5' end? Where's the 3' end? Where's the phosphodiester bond? Where's the ribose? Where's the base? Why are you majoring in MCB? No. I promise you, this is worth learning because this knowledge will shine. I want to briefly overwhelm you with the size of your genome. This picture is 4 base pairs. The string of text is 4 nucleotides. The string of text is 300 nucleotides. The human genome, of course, is 3.3 times 10 to the 9th. Put down your pencils, look up, and prepare to be awed by how much it takes to make you. This, folks, is illegible, but is actually the Declaration of Independence of this country. I thought this would be appropriate since we just celebrated President's Day. And our 3rd president, Thomas Jefferson, wrote good chunks of this. So there it goes. We hold these truths to be self-evident, that all men are created equal, they're endowed with their creator by certain unalienable rights, etc. Life, liberty, foundational statement of this country. Before you is 6,810 characters. Okay? But I told you that the human genome was 3.3 times 10 to the 9th. Okay? So if you parse the human genome into units of that size, that much text, you get 484,000 units. Okay? Your textbook, I looked, contains 1,283 pages. Okay? Let us print the human genome in your textbook with as much information as on this previous slide per page. We will need 378 copies of your textbook. I then took an old-fashioned ruler and measured it. It's 1.9 inches thick. So how tall would a stack of 378 of your textbooks be if you print that much genetic information as on the previous slide per page? It would rise to 59 feet. And that's 10.1 units of me. I am 5'11". So you need to visualize 10 people as tall as I am standing on top of one another, and then a stack of textbooks rising from the floor to the tip of the head of the topmost me, with as much information as on the previous slide per page, and you have that much information in every single one of your cells. And that's about 0.3% of it. Your textbook has this very endearingly flat representation of DNA. I want to emphasize to you that, in fact, if DNA were to look like this, it would work. Fortunately, DNA doesn't look like this. DNA is unbelievably beautiful, and I'm going to walk you through the structure in a second. But before I do that, I need to very quickly walk you through a bit of history. You've all seen this picture. This is James Watson and Francis Crick that proposed the correct model of the DNA double helix. Missing from this picture is the woman whose data were essential to the fact that Watson and Crick were able to come up with the correct model. Her picture is here. That's Rosalind Franklin. She was an X-ray crystallographer. If you Google image Rosalind Franklin, I did this a few days ago, there are all her pictures, and then the last one is that. And I have to admit how I find this very moving. I think this is the supreme honor for a scientist. Her image are her data. She was a brilliant experimentalist, an incredibly gifted X-ray crystallographer. What is X-ray crystallography? Well, I'm about to show you the method, but I'd like to show you the picture first. This was a key piece of data that she obtained, that Watson and Crick used to propose the correct model of DNA structure. There's zero doubt in my mind that she would have won the Nobel Prize. Rosalind Franklin died at age 38 of ovarian cancer. And the Nobel Prize, unfortunately, is not awarded posthumously. But, you know, in some strange way, it doesn't matter because her achievements stands next to that of Watson and Crick in the history of science. And it's a shame she wasn't there to celebrate the prize, but she is celebrated nonetheless. The technique she used is X-ray crystallography. You shine X-rays at a crystal. I don't want to take time explaining to you how X-ray crystallography works, except to encourage you to look it up on your own time. Why, for example, does it matter that the wavelength of X-rays is approximately similar to that of the size of a hydrogen atom? Why do you need a crystal? And perhaps most importantly, how do you go from this to what I'm about to show you, which is this? That's the question. Look it up on your own time. This won't be on the midterm. Let me walk you through the double helix very quickly. So the first thing to understand is, of course, DNA has two strands, just like the ones you saw a few slides ago, wound around each other in this absolutely beautiful right-handed helix. The strands themselves are held together by covalent phosphodiester bonds. This is individual strands. When they're wound together, the forces that hold them together, of course, are hydrogen bonds. And hydrogen bonds are 10 times weaker than covalent bonds, at least. And this has really interesting implications for how DNA functions. As all of you know, there are rules according to which bases on one strand pair with bases on another. Let me show you why that's remarkable. Let me just show you a base pair. That's a guanine base pairing with a cytosine. I'm hopeful you can see where the purine is and where the pyrimidine is. Let me now show you an AT base pair. And one more time. CG, TA. The overall geometry is the same. This is why the helix overall, despite the fact that the sequence is different, A, C, G, 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 C, T, A, G, G, etc., there are no bulges. The bases just fit together perfectly. What are the other two things I'd like you to know about this? Well, the first thing I'd like to point out is the two strands run in opposite directions. And I think the best way to show this is for me to show you a nucleotide distep and then maybe a nucleotide tetrastep. Let's see if this works. Here's a tetranucleotide. Let me zoom in on this a little bit. And what I want you to do is focus on this. This is the oxygen in the ring of the phosphodiester of the backbone of DNA. Notice how it's facing down. So this strand is running down. If you look at the other strand, notice how the oxygens are facing up. That strand is running up. So they are anti-parallel. Last but not least, it has a beautiful structural feature, this double helix. Namely, and I'm going to rotate it for you so you can actually appreciate it better. Nope, not the one I was going to use. Let's rotate it sideways and admire the major groove. Why is it called the major groove? Because that's the minor groove. And that's the major groove. And that's the minor groove. You get the picture. The way the strands are wound around each other, there are different size openings on the surface of the double helix. A smaller one, hello, and a larger one. So I want you to be able to look at the structure and point to me at the exam on where the major and minor grooves are. Okay, folks, I have just told you. I'll just say this very briefly. This slide lists 3 things that I encourage you with all my heart to actually read. At the very least, read The Double Helix. It's a superb book. I don't know of another book like this in the history of science. If you want a beautiful technical description of what was done, read Sir Aaron Klug's review of it. Aaron Klug was there. He was Rosalind Franklin's last collaborator, so he speaks from knowledge. It's beautifully written. And Horace Judson's Eighth Day of Creation is a marvelous narrative history of the glorious days of the 40s through the 70s. And the Nature website has a wonderful archive of all of these articles. You don't have to read any of this. I strongly recommend you read The Double Helix, but if you don't want to, that's fine. Professor Günther Stent of UC Berkeley actually edited a wonderful edition of it that I really recommend. I just told you that DNA is a right-handed double helix. The phosphate backbone is on the outside, base is on the inside, has major and minor grooves. The chains run in an anti-parallel fashion. The pairing follows Chargaff's rules. Do you need to know that term? No, but I can just tell you that that's what they're called. A with T, C with G, T with A, G with C. The strands are held together by hydrogen bonds, while the phosphodiester bond is covalent. Other than the word Chargaff, what would I like you to know from this slide? Please, everything. Please memorize this. By show of hands, how many of you know what book this is from? The Great Gatsby. Good for you. Show of hands. Somebody? The Catcher in the Rye. Good for you. Now, this is tough. Any English lit minors? If anybody knows this, I'll fall down. Ulysses. Wow. Good for you. Now, my favorite one is this. It hasn't escaped our notice that the specific pairing we have postulated immediately suggests a possible copying mechanism for the genetic material. This is the last sentence of the Watson-Crick paper. They don't talk about the genetic implications of their structure, but they did put this sentence in because they didn't want to be scooped by anybody else. The structure screams that it is built to be replicated. And that's what the genetic material should be able to do. From Kirk Douglas to Michael Douglas, pass on a copy. How does it do that? The moment the structure was proposed, everybody fell in love with it. And then people started, jumping off intellectual bridges because it wasn't clear to them how you unwind this thing. And then Matthew Michelson and Franklin Stahl did what is called the most beautiful experiment in biology. Now, you can have an argument. Is it really, really, really the most beautiful experiment? I don't want to argue about this. It is gorgeous. And I will walk you through it. It's really, really stunning. Hobbes states the problem well. Calvin goes, help me with this homework, okay? What's 6 plus 3? Hobbes, 6 plus 3, eh? Well, this one's a bit tricky. First, we call the answer why, as in, why do we care? Why should you care how DNA replicates? Intracellular processes are invisible to the naked eye, and in fact, barely visible even to the eye dressed in haute couture, namely armed with a range of next-generation tools. The Michelson-Stahl experiment is a beautiful example of how biologists study and understand a cellular process by observing its mechanics indirectly. Neither Michelson nor Stahl went inside the nucleus and looked with their eyes at the replication fork. Your textbook explains this well. I am expecting you to spend a substantial amount of time studying and understanding this experiment in complete detail, discussing it with your GSIs, coming to me to office hours. I can only have so much time to explain that what your book explains well. I do want to explain the problem. Your textbook has this nice color schematic in which you could imagine 3 modalities of DNA replication. First, semi-conservative. The 2 strands unwind. Each of the strands becomes the basis for synthesis of a complementary strand, and then this hybrid molecule exists. Alternatively, maybe the DNA doesn't unwind and just builds another one next to it. You, make a copy of me. Finally, there could be dispersive replication, where just random weirdness occurs, more or less. I think the textbook does an injustice by giving you the conclusion. You must understand that chemically, DNA is all the same. The left strand and the right strand, the top strand, the bottom strand. When Michelson and Stahl walked up to this problem, this is what the world looked like. It was black and white. Because all DNA strands are the same, there was no way to tell. Well, you get DNA from DNA. Great. Which of the 3 models is this? They found a way to color code the DNA in living cells. That's Dr. Matt Michelson in front of a Model E analytical ultracentrifuge. And that's the time when this experiment was done, 1956. And that's Dr. Stahl with his wife. They are very cool, Michelson and Stahl. And in fact, I urge you with all my heart to go listen to Matt Michelson explain his own experiment. There's this great website, and he talks about it. He shows his data, and it's really moving. His voice starts to break when he shows the primary piece of data. And believe me, I go, oh my God. Seriously. So the bottom line is, I suspect that many of you have already studied this experiment in one shape or form, So my plan is to go through this relatively quickly. The fundamental idea is an unbelievably straightforward one, is to differentially label original and newly synthesized DNA strands. Differentially label with what? A heavier or a lighter isotope of nitrogen. And how do you then distinguish between whether or not an original molecule makes a perfect copy or whether there is a hybrid? You use a density gradient. Namely, you use a tool that allows you to separate lighter DNA from heavier DNA. And as your textbook explains nicely, depending on which of these three models is correct, you would expect to see one of three different patterns after you grow the bacteria. This was done in bacteria for one and then 2 generations in a different kind of isotope. So you begin by growing bacteria in isotope type 1, let's say the heavy one. And then you grow them for 1 generation in light isotope and then for another generation in light isotope. And as your textbook explains well, the patterns you will see from a density perspective of these parental and daughter molecules will be different. Very briefly, beauty is in the eye of the beholder. This is one of the most beautiful things I've ever seen. This is Gian Lorenzo Bernini's statue of Apollo and Daphne. But I'm prepared to accept the fact that I'm, biased. I was raised with Western values. I'm accepting the fact that some of you will look at this and go, huh. This, however, is universally beautiful. These are Michelson and Stahl's data, the primary data. This is heavy DNA after multiple generations of being grown in heavy isotope. And then after 1 generation of being grown in light isotope, you see a hybrid band. Oh, it's of intermediate density. But if the model is true, then after 2 generations, you should see exclusively light DNA and 1 hybrid DNA, and that is what you see. I don't know of a single biologist who, when looking at this, wouldn't go, oh, that's so beautiful. And I respect the fact that you go, those are weird-looking bands on a thing. Trust me, if you join us, you will agree with us. DNA replicates semi-conservatively. I promised you a movie, and with that, I'll let you go. I want this movie to haunt you through the rest of the weekend. I told you we're going to learn forensics, and on Monday we'll also learn about mitosis. Don't go away. I promise. This is worth it. I got this movie from Quorum Technologies. Thank you to them for providing it to me. It's the highest resolution movie I have ever seen of mitosis in the early Drosophila embryo. Are you still with me? After fertilization in Drosophila, in fruit flies, but not in humans, all the nuclei live in the same cytoplasm. So they follow the same cell cycle control signals. And without absolutely any further ado, oh, what's happened here? Okay, let's reopen it. Stay with me. I promise you, this is worth it. Hey, Anna, can you kill all the lights, please? The circles are the nuclei. You're about to see the chromosomes. They all obey the same cytoplasmic signal. Individual cell membranes haven't formed yet, which is why they do this. I think they will do this one last time, and then each nucleus will form its own membrane and will stop listening to what the others say. I think there's one more. Have a nice weekend.